Today we are starting with a new video series, reading a new book that is related to our world now theme, that is coronavirus. The book is for the SARS, that is a pandemic in 2003, but to know very well what coronavirus is, let's begin with it. SARS and other coronaviruses laboratory protocols. The book is done in San Francisco. With the first title now, a pan-coronavirus RT-PCR assay for detection of all known coronaviruses. Introduction. At present, viral culture is the gold standard for laboratory diagnosis of respiratory infections. Since coronaviruses are very difficult to grow in cell culture, accurate and sensitive diagnoses are not feasible by this technique. To overcome the lack of sensitivity and to obtain rapid diagnostic results, more sensitive molecular methods for the detection of human coronaviruses (nCOVs) have been developed, including reverse transcriptase polymerase chain reaction (RT-PCR), nested RT-PCR, and recently real-time RT-PCR. Nevertheless, nCOVs are not often diagnosed in clinical laboratories, although the SARS epidemic and the identification of nCOV-19 drew attention to the clinical relevance of nCOVs. Multiplex RT-PCRs for the detection of common respiratory viruses in clinical specimens have now been supplemented with primer pairs for amplification of nCOV-OC43, 229E, and NL63 in order to allow the detection of all known coronaviruses in one assay. Consensus RT-PCRs have been developed based on alignment of conserved genome regions of several coronaviruses. However, the recent identification of nCOV-NL63 demonstrated that this novel coronavirus could not be amplified by the consensus RT-PCR described by Stevenson and colleagues, which might be explained by the presence of several mismatches in the primer sequences. We designed a novel pan-coronavirus RT-PCR in which we modified the coronavirus consensus RT-PCR primers based on alignment on the nCOV-NL63 sequence and the corresponding sequences of 13 other coronaviruses. Theoretically, this pan-coronavirus RT-PCR should amplify a 251 BP fragment of the polymerase gene of all coronaviruses known at present, and we tested this experimentally for the 5 known human coronavirus nCOV-NL63, 229E, OC43, HKU1, and SARS-CoV, and 3 animal coronaviruses, FEV, and MHV. The sensitivity of the pan-coronavirus RT-PCR was found to be lower than for a non-degenerate RT-PCR, which can be explained by the high level of degeneracy of the pan-coronavirus primers. Nevertheless, this pan-coronavirus assay is a useful tool for screening sample collections for the presence of all known and potentially yet unknown coronaviruses. That is the case today. 2 materials. 1. Sample handling and RNA extraction. 1. Viral transport medium VTM BD diagnostic systems USA or phosphate buffered solution BPS. Store at room temperature. Filter minisart plus 045 micrometer Sartorius Vivasens AG, 2 ml syringe without a needle, glass pierce 5 mm diameter. Merck MDA, VWR Belgium, Quam Viral RNA Mini Kit, Kruingen, Netherlands, stored at room temperature except for the AVL lysis buffer, which should be stored at 4 degrees Celsius after addition of carrier RNA. 2. Pan-coronavirus one-step RT-PCR. The first primers, forward primer and reverse primer. The primers are prepared in a stock solution of 100 micrometer. A working solution of 15 micrometer is made for use in the RT-PCR stored at -20 degrees Celsius until use. Then the next margins and steps are plus scientifically and medicine people that know about this. We will go into very sensitive and very explaining the situation of this coronavirus, then do another pages. We'll see another pages that is simplified, and this is the beginning of the book. We'll go into the topic. We can do that. Yes, coronaviruses are a group of enveloped positive-strand single-stranded RNA viruses with a corona-like morphology. These viruses have genome size ranging from 28 to 32 kb, which makes them the biggest among the RNA viruses. The genomes contain 5 major open-reading frames that encode the replicase polyproteins ORF1A and ORF1AB, spike S, envelope E, membrane, and nucleocapsid proteins in that order. Based on antigenic and genetic analysis, coronaviruses can be subdivided into 3 groups. Coronaviruses from group 1 and 2 have been found to infect mammals. By contrast, avian species are known to be the natural hosts for group 3 viruses. Most coronaviruses are pathogenic to their hosts. Animal coronaviruses are known to cause respiratory, gastrointestinal, neurological, and hepatic diseases. This is the main places that the coronavirus is hostile and pathogenic in the human being organism. An explanation to this phrase. By contrast, with the exception of SARS coronavirus, in the case of healthy individuals, all previously known human coronaviruses were generally only associated with mild respiratory and gastrointestinal diseases. The discovery of a novel coronavirus as the cause of SARS led to a resurgence in interest in these viruses and to the discovery of other novel coronaviruses in humans, namely NL63 and HKU, the carnation of SARS-like coronaviruses in pound civets and other small mammals in live game animal markets, prompted intensive surveillance for coronaviruses in wild animals. Following upon the interesting discovery, several novel coronaviruses were identified in the mammalian and avian species. In particular, our initial studies first revealed that baits are important reservoirs for coronaviruses. By screening respiratory and fecal samples collected from a wide range of animals, we identified the first coronavirus in baits. Subsequent studies by ourselves and others also indicated that there are a number of group 1 and group 2 viruses circulating in bat species. In particular, a SARS-like coronavirus was found in 2 of these studies independently. These results suggest that baits might play a key role in the evolution of coronaviruses. Like our days, it's the bait that is the cause of this coronavirus. It's the first home of this virus. It was the bait. The main problem is remaining from 2003 with SARS. Then let's resume. Here we describe the procedures that were used in our virus surveillance studies, and all that we can read for this book is being done. Now that's all for today's topic, coronavirus, that we can extract from this book. I hope you enjoy reading.